Okay, welcome to the plasma membrane. Like we said last week, the plasma membrane is one of our organelles. It's found in prokaryotes, eukaryotes, plant, animal cells, and it is so complex that it gets its own chapter. So just as a reminder, the plasma membrane is what surrounds the cytoplasm. It's going to determine the nature of how the cell interacts with its environment. Because it's selectively permeable and it allows certain things to come into the cell and certain things to leave the cell. It's flexible in some places of the membrane, rigid in others, but the rigidity and flexibility will depend on the type of cell. It will also carry cellular recognition markers to allow the determination of self from non-self. So we would be able to tell foreign invaders from cells that are normally within our bodies. So first we'll start off with the fluid mosaic model. It was discovered in the 1890s. Its chemical components were discovered in 1915. In 1935, Davison and Danieli based their model of the plasma membrane off of a railroad track appearance that they saw on electron micrographs. They determined from that the plasma membrane resembles a sandwich, which it kind of does. In 1972, Singer and Nicholson came up with the fluid mosaic model, and the fluid mosaic model is kind of what we use as a standard for what the plasma membrane is like today. So it's just like it sounds. The plasma membrane has this fluidity to it, but it also has mosaic components, all of these proteins and lipids and carbohydrates that are embedded within it, almost like a jello with pieces of fruit and whatnot stuck in it in different places. It's fluid mosaic. It gives the membrane its fluidity, but also its structure. So the components of the plasma membrane will vary depending on the cell type. Lipids make up about 40% of the plasma membrane, and they're made up of phospholipids and cholesterol mainly. Proteins make up about 50% of the plasma membrane, with carbs making up about 10%. And that's composed of glycoproteins and glycolipids, and we'll talk about those. So remember that phospholipids have these hydrophilic and hydrophobic components. So the basic structure of this phospholipid bilayer, this plasma membrane, is going to have these hydrophilic heads on the outside and hydrophobic tails on the inside. So you can see it kind of does look like a sandwich. These phospholipids are vital to the function of the plasma membrane, as it keeps polar molecules out unless they are able to pass through via a carrier or a channel, and we'll talk about those in a few minutes. And it allows nonpolar molecules to just pass through. Once phospholipids are heated in an aqueous solution, they form small spheres and droplets, known as micelles and liposomes. So we talked a little bit about micelles last week. Micelles are these spheres of phospholipids that are formed. The phospholipids that form the spheres in micelles are just a single layer, whereas liposomes, those are the droplets, they form a bilayer sphere. So let's talk about the proteins that are in the plasma membrane. There are two main types, integral proteins and peripheral proteins. Integral proteins are, as the name suggests, integral. So they travel all the way through the plasma membrane from the extracellular portion to the intracellular portion. They are integral in the way that if you were to try to remove them from the membrane, they would completely destroy or damage the plasma membrane beyond repair. They have some characteristics to help determine self from non-self, as well as some enzymatic activity in certain integral proteins. Some act as channels or carrier proteins, and we will talk about those soon. Those are also known as integrins, and in this picture on the side, you can see that an alpha helix, a collection of alpha helices, or a beta pleated sheets can form the structure of these integral proteins. Peripheral proteins are known as cell-specific proteins. They're found on either the exterior or the interior surface. There will be multiple peripheral proteins on a plasma membrane, where some will be interior, some will be exterior, depending on their functions. They can also be attached to integral proteins or phospholipids. They also serve as enzymes, could determine self from non-self, or attachment sites for certain molecules. They are not as integral as integral proteins are, in the sense that you could remove peripheral proteins without them damaging the plasma membrane itself. Carbohydrates will always be on the exterior surface of the plasma membrane, and they will be either bound to proteins or lipids in the form of glycoproteins or glycolipids. Together, that forms the glycocalyx. The glycocalyx is a sugar coating. It's hydrophilic and tends to attract a lot of water into the plasma membrane. It helps to form cellular attachments with other cells to form tissues. And some viruses tend to have a similar glycoprotein-glycolipid structure on the outer surface, which can lead to other cells having difficulty recognizing them, and difficulty then getting rid of these viruses. So like I said, with the viruses, cells have certain binding sites that are specific to certain viruses, and antibodies will recognize the surface markers of these viruses and attack them to rid of them. But sometimes the glycoprotein-glycolipid patterns on these viruses so closely resemble our cells that it's difficult for these antibodies to attack them. Oftentimes with some of our trickier viruses like HIV, they have recognition sites that mutate, so it makes them even harder to kill. So the membrane, like we said before, is both fluid and rigid. Due to this fluidity and rigidity, it can burst if under the improper conditions. So there are two main factors that maintain the fluidity of the membrane: the nature of the phospholipids and the cholesterol. With the phospholipids, it's mostly the bond patterns that's going to help maintain that fluidity. The straight tails of the phospholipids, the dense rigid membranes in lower temperatures, and the space between cells at higher temperatures. With cholesterol, it will dampen the temperature effects on membranes and function as a buffer so that extreme temperatures do not affect the membrane. Of course, this is only to certain extremes. Past physiologically possible temperatures for humans or animals, those cell membranes will end up freezing or bursting depending on the conditions. So here is a recap of the components and locations. It's a really nice table to look at. So we've said in the past that plasma membranes are selectively permeable. The cell could be destroyed without this because it allows for certain nutrients to come in, waste to be excreted, and if just an unlimited amount of substances or ions were allowed to pass through, the cell would not be able to function. So transport across the membrane occurs via 3 different processes: passive transport, facilitated transport, and active transport. Passive and facilitated transport do not require any energy to be used, but active does. So all of the components of the plasma membrane are going to contribute to this selective permeability. Non-polar lipid soluble substances are allowed to pass through, and some will need transport assistance. So we'll talk about that transport assistance. Diffusion is our first form of passive transport that we will talk about, meaning that it does not require energy. So with passive transport, solutes are going to move from an area of high concentration to low concentration. That's along a gradient. So you can see here that we have our lipid bilayer. We have a high concentration of solutes in the extracellular fluid and not many on the intracellular fluid. So these solutes will want to diffuse through the plasma membrane and into the cytoplasm until they reach a sort of equilibrium. They will always want to balance out, so everything will travel toward equilibrium. It'll be a dynamic equilibrium with some leaving, some coming in, but they always want to kind of balance out as much as they possibly can. There are some factors that affect diffusion. The greater the difference in the concentration gradient, for example, the more solutes that are on one side rather than the other, makes for more rapid diffusion. The heavier the molecules that are diffusing, the slower the diffusion will take place. The higher the temperature, the greater the rate of diffusion. The higher the density, the lower the rate of diffusion. So we can kind of look at that with somebody who's dehydrated, because their body cells will lose water, diffusion will lower or decrease, and cells will stop functioning. Nonpolar lipid-soluble molecules will have a higher rate of diffusion. Greater surface area will increase the rate of diffusion. A thicker membrane will decrease the rate of diffusion. And a greater distance that has to be traveled across this membrane will lower the rate of diffusion as well. Filtration is a variation of diffusion. It's very similar of diffusion, where it's moving toward that concentration gradient to kind of balance out these solutes. Infiltration, oftentimes it's due to some sort of pressure. For example, in the kidneys, blood pressure is facilitating that filtration. So next we have facilitated transport, or facilitated diffusion. And this is diffusion across a membrane with the help of proteins, either channels or carrier proteins. So with channels, we have transport proteins or channel proteins, and transport proteins can function as either carriers or channels. Channels are exposed to both the intra and extracellular fluids because they are integral proteins, so they run through the whole length of the plasma membrane. It's much quicker for diffusion to take place if they go through channels. And there's usually a central channel in plasma membranes that is hydrophilic, that allows for the quick passage of polar molecules. Aquaporins are also found as channels in the plasma membrane to allow the quick passage of water. And some channels can be gated, meaning that they can turn on and off. Most of the examples are ion-gated channels, so if there's enough of the ions on the inside of the cell in the intracellular fluid, then the gate will be closed. It will stay closed until more ions are needed. Examples are in the renal tubules, also in nerves and muscles, where they need certain ions in order to perform their functions. Similarly, carrier proteins are going to bind a substrate. They will change their shape, and then they will move the molecule inside the cell. Typically, they are specific to their substance. So you can see here that these molecules are binding. The protein changes shape and then allows for that molecule to enter into the cell. One example of this is the GLUT transporters, or the GLUT proteins. These are specific to glucose molecules, and they function when insulin triggers them to. So when insulin is circulating in the bloodstream to lower the blood sugar, insulin tells these GLUT transporters to open and allow more glucose into the cell, so that they're within the cell rather than free-floating in the blood. Similarly, osmosis is going to be the movement of specifically water across a semi-permeable membrane. While this will seem like the opposite of what we've just been doing, where water will be moving from high to low concentrations of water, it's still moving from low to high concentrations of solutes. So we're thinking of this red dotted line as the membrane. Notice in this beaker, on the left-hand side of the membrane, there are less solutes than on the right side. So if there's a lot of solutes on the right side, that there's less water, because these solutes are displacing the amount of water that can be present. So water will want to travel from the left side of the membrane to the right side of the membrane, in this case, to balance out the amount of water on both sides. Although this doesn't look balanced, it accounts for the presence of the solutes that's displacing the amount of water on this side. So the water will always want to move toward the area of higher concentration of solutes and lower concentration of water. Tonicity is going to describe how an extracellular solution can affect a cell's volume via osmosis. This will correlate directly with osmolarity, which is the cell's total solute concentration. So solutions can either be hypotonic, hypertonic, or isotonic. In hypotonic solutions, the extracellular fluid of the cell has a lower osmolarity than the fluid inside the cell. So the osmolarity is the solute concentration. So if there's less solutes on the outside of the cell, more solutes on the inside of the cell, water will want to flow toward the area of more solute concentration, higher solute concentration. So water will rush into the cell, and in this case we're using the example of red blood cells. The blood cells would burst because of the increase of water being rushed into the cell, and this is called lysis. Contrarily, if the extracellular fluid has a higher osmolarity than the inside of the cell, and there's more solutes on the outside, less solutes on the inside, water will want to rush out of the cell and into the extracellular environment, causing the cells to shrivel up or crenate. And in isotonic solutions, the extracellular fluid and the cell have the same osmolarity, so there's no net movement of water into or out of the cell. That's not to say that there's no movement of water. Water's still coming in and going out, but it's maintaining a dynamic equilibrium. So many living things have osmoregulation. They're able to control the effects of osmosis so that they don't have this lysis and crenation happening in their cells. In organisms that have cell walls, the cell walls help to protect the cells from lysis when they're in hypotonic solutions. Plants can undergo plasmolysis if the extracellular fluid of the plant becomes hypertonic. Then water will rush out of the plant cells into the extracellular fluid. The cell membrane will detach from the cell wall and the plant will wilt. In paramecia and amoebas, they have special organelles called contractile vacuoles. In contractile vacuoles, they collect excess water and pump it out to prevent themselves from bursting because they are unicellular organisms. Some marine invertebrates match the salt level to be isotonic with their environments so that their cells can also maintain their stability. And some fish use their metabolic energy. Most use about 5% of their metabolic energy. Of all the ATP they are creating for energy for any of their processes in their body, they're using 5% of that to be able to maintain their osmolarity and pumping the right amount of salt into their bodies. And that will vary depending on the type of fish. This week's discussion board is actually about osmoregulation in fish and organisms that live in both salt and freshwater environments. So make sure that you take a look at that. It's very interesting. And we'll pick up where we left off here next week.